RNA synthesis and processing are central to cellular function, as they are the key steps in the flow of genetic information from DNA to protein. The process begins with transcription, where an RNA molecule is synthesized from a DNA template, and continues with various processing steps that convert the primary RNA transcript into a mature, functional RNA. The main types of RNA involved are messenger RNA, mRNA, ribosomal RNA, rRNA, and transport RNA, all of which undergo specific processing steps before they perform their roles in protein synthesis. In this lecture, we will cover both RNA synthesis, transcription, and processing in detail, incorporating key points related to the CSIRNet JRF Life Sciences syllabus. 1. RNA synthesis, transcription. Transcription is the first step in gene expression, during which a particular segment of DNA is copied into RNA by RNA polymerase. The process involves several stages: initiation, elongation, and termination. Initiation. Transcription begins when RNA polymerase binds to a specific sequence of DNA called the promoter region. The promoter is recognized by the sigma factor in prokaryotes or transcription factors in eukaryotes, which guide RNA polymerase to the correct starting point. In eukaryotes, transcription begins with the binding of the transcription initiation complex, which includes transcription factors such as TFID, which binds to the top box, and RNA polymerase II. Elongation. During elongation, RNA polymerase moves along the DNA template strand in a 3' to 5' direction, synthesizing the RNA in the 5' to 3' direction. As the polymerase moves, the DNA template unwinds, and the newly synthesized RNA is elongated by the addition of ribonucleotides complementary to the DNA template. Termination. The termination of transcription occurs when RNA polymerase reaches a termination signal in the DNA. In prokaryotes, this can involve a terminator sequence that causes the You said, continue ChatGPT said. RNA polymerase to dissociate from the DNA, releasing the newly synthesized RNA transcript. In eukaryotes, termination is more complex and involves additional factors like the cleavage of the RNA transcript followed by polyadenylation, the addition of a poly A tail. In eukaryotes, transcription occurs in the nucleus, and there are three main types of RNA polymerases. RNA polymerase I synthesizes rRNA except 5sRRNA. RNA polymerase II synthesizes mRNA, snRNA, and some RNAs. RNA polymerase III synthesizes tRNA, 5sRRNA, and other small RNAs. Prokaryotes only have a single RNA polymerase responsible for the transcription of all types of RNA. The process is relatively simpler, but it still involves similar steps of initiation, elongation, and termination. 2. RNA processing in eukaryotes. After transcription in eukaryotic cells, the primary RNA transcript, also known as pre-mRNA, undergoes several modifications before it becomes a mature mRNA molecule capable of being translated into a protein. These processing steps include capping, splicing, and polyadenylation. Capping. Once the RNA transcript is synthesized, a 5' cap is added to the beginning of the mRNA. This cap is a modified guanine nucleotide, 7-methylguanosine, that protects the mRNA from degradation and aids in the initiation of translation. The 5' cap also facilitates mRNA export from the nucleus to the cytoplasm. Splicing. Eukaryotic genes often contain non-coding sequences called introns, which must be removed from the pre-mRNA. The remaining coding sequences, exons, are joined together in a process called splicing. This is carried out by a complex called the spliceosome, which recognizes specific sequences at the junctions of introns and exons. Alternative splicing allows a single gene to produce multiple mRNA variants, contributing to proteome diversity. Polyadenylation. After transcription is completed, a poly tail is added to the 3' end of the mRNA. This tail consists of a long chain of adenine nucleotides, typically 200 to 250, and plays several important roles, including mRNA stability, protection from exonucleases, and facilitation of mRNA export from the nucleus. Polyadenylation is carried out by the polypolymerase enzyme after the mRNA is cleaved at a specific site. 3. RNA editing and modifications. In addition to the primary steps of RNA synthesis and processing, there are other forms of RNA modifications and editing. For example, in RNA editing, specific nucleotides within the RNA sequence can be altered post-transcriptionally. One common example is adenosine to inosine editing, which occurs in certain mRNAs and can affect the coding potential of the RNA. 4. Differences between prokaryotic and eukaryotic RNA synthesis and processing. Prokaryotic transcription is relatively straightforward. There is no separation between transcription and translation. Translation can begin on the mRNA even while it is being transcribed. There are no introns in prokaryotic genes, so splicing is not necessary. Eukaryotic transcription is more complex. Transcription occurs in the nucleus, and mRNA processing occurs before the mRNA is transported to the cytoplasm. Eukaryotic mRNAs are typically much larger, containing both exons and introns, requiring more extensive processing, capping, splicing, and polyadenylation. 5. Key regulatory elements in transcription and processing. Several regulatory elements control RNA synthesis and processing. Promoters and enhancers. These DNA sequences regulate the initiation of transcription. Promoters are located near the start site of transcription, while enhancers can be located further away and help enhance the transcription rate. Transcription factors. Proteins that bind to specific DNA sequences near the promoter to initiate transcription. In eukaryotes, the interaction of transcription factors and RNA polymerase II is crucial for accurate transcription initiation. RNA binding proteins. These proteins interact with nascent RNA molecules and participate in processes like splicing, transport, and stability. For example, SR proteins are involved in the splicing process, while HNRNPs, heterogeneous nuclear ribonucleoproteins, play a role in RNA processing and stability. Previous year questions. Question. CSIRNET 2021. Which of the following modifications occur in eukaryotic pre-mRNA during transcription? A. Methylation of the guanine base at the 5' end. B. Addition of a poly tail at the 5' end. C. Splicing of exons. D. Deletion of exons. Answer. A. Methylation of the guanine base at the 5' end. This modification is a 5' cap, which is crucial for mRNA stability and translation initiation. Question. CSIRNET 2020. Which of the following RNA polymerases is responsible for the transcription of mRNA in eukaryotes? A. RNA polymerase I. B. RNA polymerase II. C. RNA polymerase III. D. All of the above. Answer. B. RNA polymerase II. RNA polymerase II is responsible for synthesizing mRNA in eukaryotes. Question. CSIRNET 2019. What is the function of the 5' cap added to eukaryotic mRNA? A. To protect the RNA from exonuclease degradation. B. To enhance the translation efficiency. C. To facilitate splicing of introns. D. Both A and B. Answer. D. Both A and B. The 5' cap protects the mRNA from exonuclease degradation and aids in translation efficiency by assisting ribosome binding. Conclusion. RNA synthesis and processing are fundamental processes that gene express. In eukaryotes, transcription is followed by significant processing steps, including capping, splicing, and polyadenylation, all of which are essential for the production of functional mRNA. Understanding the molecular mechanisms involved in RNA synthesis and processing is critical for answering questions related to gene expression regulation, and this topic is frequently tested in the CSIRNet JRF Life Sciences exam. Make sure to review the key elements, such as the types of RNA polymerases, the function of the 5' cap, splicing mechanisms, and polyadenylation, as these are frequently assessed. Let me know if you'd like to proceed with another topic or need further clarification. You said. Transcription factors. ChatGPT said. Transcription factors CSI RNet JRF Life Sciences. Transcription factors TFs are essential regulatory proteins that control the process of transcription by facilitating or inhibiting the binding of RNA polymerase to specific genes. They are key players in gene expression regulation, determining when and how much a gene is transcribed into RNA. Transcription factors function by recognizing specific DNA sequences called promoter regions or enhancer sequences in the genome, and they play a vital role in the activation or repression of gene expression in both prokaryotic and eukaryotic organisms. This lecture will provide an in-depth explanation of transcription factors, their types, mechanisms of action, and their role in regulating gene expression. The following aspects will be covered. Overview of transcription factors. Classification of transcription factors. Mechanisms of action. Cofactors and company regulators. Transcription factors in eukaryotes versus prokaryotes. Regulation of transcription factors. Previous year questions. 1. Overview of transcription factors. Transcription factors are proteins that bind to specific DNA sequences and are critical for the regulation of gene expression. They work by either recruiting RNA polymerase to the gene's promoter or by influencing the activity of RNA polymerase once it is bound to DNA. In eukaryotes, transcription factors typically function in conjunction with RNA polymerase II, which is responsible for transcribing mRNA. These transcription factors are part of larger complexes and often interact with other proteins to modify chromatin structure or facilitate transcriptional initiation. In prokaryotes, transcription factors interact with the RNA polymerase core enzyme to guide it to specific promoters, ensuring proper transcription initiation. 2. Classification of transcription factors. Transcription factors can be classified into two broad categories based on their function. General transcription factors (GTFs). These are required for the transcription of most genes and are involved in the formation of the transcription initiation complex. They help RNA polymerase bind to the promoter. GTFs include proteins such as TFID, TFIIA, TFIIB, TFIIF, TFIIE, and TFIIH, which are essential for the initiation of transcription by RNA polymerase II in eukaryotes. Regulatory transcription factors. These factors control the transcription of specific genes in response to various signals. They bind to enhancers, silencers, or promoters to regulate transcriptions. Some examples include activators, which enhance gene expression, and repressors, which inhibit gene expression. Regulatory transcription factors are often tissue-specific and may respond to signals such as hormones or environmental factors. Within regulatory transcription factors, there are various families based on their structural domains and DNA binding motifs, such as helix turn helix, zinc finger, leucine zipper, basic helix loop helix BHLH. 3. Mechanisms of action. Transcription factors regulate gene expression through various mechanisms. Binding to promoter regions. Many transcription factors bind to specific sequences in the promoter region of genes, near the transcription start site. These sequences, such as the TATA box recognized by TFID in eukaryotes or the Pritnell box in prokaryotes, are essential for the initiation of transcription. Recruiting cofactors. Transcription factors do not always act alone. They often recruit cofactors such as coactivators, which enhance gene expression, or core repressors, which suppress transcription. Coactivators and core repressors help remodel chromatin structure, modify histones, or facilitate RNA polymerase's ability to transcribe DNA. Interacting with chromatin. Transcription factors can alter the structure of chromatin by interacting with chromatin remodeling complexes or histone modifying enzymes, leading to either the opening or closing of chromatin regions. Open chromatin is more accessible for transcription, whereas condensed chromatin inhibits transcription. Modifying RNA polymerase activity. Some transcription factors can also regulate the activity of RNA polymerase itself, either by modifying the polymerase or interacting with elongation factors to enhance or inhibit transcription elongation. 4. Cofactors and company regulators. Cofactors and co-regulators are additional proteins that do not bind directly to DNA but assist transcription factors in regulating gene expression. They can be classified into two categories. Coactivators. These enhance the transcriptional activity of transcription factors. Examples include CBP P300 and mediator complex, which help recruit the RNA polymerase complex and chromatin modifying enzymes to the promoter. Core repressors. These inhibit transcription by interacting with transcription factors and preventing the formation of an active transcription initiation complex. NCOR and SMRT are examples of core repressors that recruit histone deacetylases (HDACs) to compact the chromatin and inhibit transcription. 5. Transcription factors in eukaryotes versus prokaryotes. Eukaryotic transcription factors. In eukaryotes, transcription is more complex due to the presence of introns, exons, and chromatin. Transcription factors in eukaryotes require the formation of large complexes that interact with other regulatory proteins to initiate and regulate transcription. These factors also influence chromatin remodeling and RNA processing. Prokaryotic transcription factors. Prokaryotes have simpler transcriptional regulation. The RNA polymerase core enzyme, aided by sigma factors, binds to the promoter regions to initiate transcription. The sigma factors allow the polymerase to recognize specific promoters and direct transcription accordingly. In prokaryotes, transcription factors tend to act at the level of promoter recognition and help modulate transcription based on environmental cues. 6. Regulation of transcription factors. Transcription factors themselves are often regulated by a variety of mechanisms. Post-translational modifications. Transcription factors can be activated or inhibited by phosphorylation, acetylation, ubiquitination, or other modifications. For instance, the phosphorylation of transcription factors can influence their DNA binding ability or their interactions with cofactors. Signal transduction pathways. External signals such as hormones, growth factors, or stress conditions can activate or inhibit transcription factors. For example, the nuclear factor kappa B NF-kappa B pathway is activated in response to inflammatory signals, leading to the activation of transcription factors that regulate immune response genes. Feedback mechanisms. Transcription factors can be part of feedback loops. For instance, some transcription factors can regulate their own expression by binding to the promoter regions or interacting with other regulatory elements. Previous year questions. Question CSIR NET 2022. Which of the following transcription factors is involved in the initiation of transcription by RNA polymerase II in eukaryotes? A. TFID. B. TFIIF. C. TFIIH. D. All of the above. Answer. D. All of the above. TFIID binds to the TATA box. TFIIF helps stabilize RNA polymerase binding, and TFIIH has helicase activity and phosphorylates the RNA polymerase tail, all of which are required for transcription initiation. Question CSIR NET 2021. The basic helix loop helix BHLH motif is a feature of which type of transcription factors? A. Repressors. B. Activators. C. Both. D. None. Answer. B. Activators. The BHLH motif is commonly found in transcription factors that act as activators, especially in the regulation of developmental processes and cell differentiation. Question CSIR NET 2020. Which of the following statements is true about transcription factors? A. They only bind to enhancer regions in DNA. B. They regulate transcription by recruiting RNA polymerase to the promoter. C. They are always repressors. D. They are only involved in prokaryotic gene expression. Answer. B. They regulate transcription by recruiting RNA polymerase promoter. Transcription factors play a critical role in guiding RNA polymerase to the gene's promoter and modulating the transcription process. Conclusion. Transcription factors are fundamental in regulating gene expression in both prokaryotes and eukaryotes. Their ability to bind to specific DNA sequences, recruit cofactors, and interact with chromatin remodeling complexes ensures that genes are expressed at the right time and in the correct cell types. Understanding the various mechanisms by which transcription factors regulate gene expression is crucial for tackling questions in the CSIR-NET JRF Life Sciences exam. Focus on key transcription factors, their families, and regulatory mechanisms to ensure a strong grasp of the topic. Let me know if you'd like to explore another topic. You said. Transcription factors machinery ChatGPT said. Transcription factor machinery CSIR-NET JRF Life Sciences. Transcription is a highly regulated process that requires the coordinated action of multiple proteins and complexes. The machinery involved in transcription includes not only the transcription factors themselves but also a variety of additional components that enable the efficient and precise transcription of genes. This machinery facilitates the binding of RNA polymerase to the DNA template, the unwinding of the DNA helix, and the initiation, elongation, and termination of RNA synthesis. The role of transcription factors is central to this process, but they work together with other essential proteins and enzymes to ensure proper gene expression. This lecture will discuss the transcription factor machinery, focusing on the components involved in the initiation and regulation of transcription. The following points will be covered. Transcription machinery overview. RNA polymerase and its subunits. General transcription factors, GTFs. Mediator complex. Chromatin remodeling complexes. Co-activators and company repressors. Post-translational modifications and regulation. Previous year questions. 1. Transcription machinery overview. Transcription machinery consists of a core set of proteins and complexes that work together to initiate and regulate the transcription of DNA into RNA. In eukaryotic cells, this machinery is especially complex due to the need to overcome chromatin compaction and the involvement of additional regulatory proteins. The key components of the transcription machinery include RNA polymerase II for mRNA synthesis in eukaryotes. General transcription factors, GTFs. Chromatin remodeling complexes. Co-activators and co-repressors. Mediator complex. Together, these components ensure that transcription is not only initiated correctly but is also regulated in response to cellular signals, ensuring proper gene expression patterns. 2. RNA polymerase and its subunits. In eukaryotes, the transcription machinery is primarily driven by RNA polymerase II, which is responsible for transcribing protein coding genes into mRNA. RNA polymerase II itself is a large enzyme made up of multiple subunits. In its inactive state, RNA polymerase II requires the assistance of general transcription factors, GTFs, to initiate transcription. Key subunits of RNA polymerase II include RPB1, largest subunit. This subunit is responsible for the catalytic activity of the polymerase. RPB2, it helps form the catalytic core of the enzyme. RPB3 to RPB12, these subunits help with structural integrity and the proper functioning of the polymerase. Once the transcription machinery is assembled at the promoter region, RNA polymerase II begins RNA synthesis after the GTFs and other cofactors have set the stage for transcription initiation. 3. General transcription factors, GTFs. General transcription factors, GTFs, are proteins required by RNA polymerase II to initiate transcription in eukaryotes. These factors are essential for the formation of the pre-initiation complex, PIC, which is a multi-protein assembly of the gene's promoter that enables transcription initiation. The key GTFs include TFIID. This is the first GTF to bind the promoter. It is a complex of the TBP-TATA binding protein and several TAS It is a complex of the TBP-TATA binding protein and several TAS TBP-associated factors that recognize the TATA box and initiate the assembly of the pre-initiation complex. TFIIA. This factor stabilizes the binding of TFIID to the promoter and helps recruit additional GTFs. TFIIB. It recruits RNA polymerase II to the promoter and stabilizes the complex. TFIIB also helps in selecting the transcription start site. TFIIF. This factor helps stabilize RNA polymerase II binding to the promoter and facilitates the transition from initiation to elongation. TFIIE. It recruits and activates TFIIH, which has helicase activity and phosphorylates the RNA polymerase II CTD C-terminal domain. TFIIH. This GTF is crucial for promoter opening and unwinding of the DNA to allow transcription to begin. It also phosphorylates the C-terminal domain of RNA polymerase II, facilitating the transition from initiation to elongation. 4. Mediator complex. The mediator complex is a large multiprotein complex that acts as an intermediary between transcription factors and RNA polymerase II. It serves as a bridge, transmitting signals from the regulatory transcription factors to RNA polymerase II. The mediator complex is essential for the activation of transcription and the recruitment of additional co-activators and chromatin remodeling factors. Mediator functions in several ways. It facilitates the interaction between activator proteins and the basal transcription machinery. It helps the transition from transcription initiation to elongation. It integrates signals from various transcription factors, ensuring coordinated gene expression. 5. Chromatin remodeling complexes. Since DNA is packaged into chromatin in eukaryotic cells, the transcription machinery must overcome the structural barriers posed by the chromatin. Chromatin remodeling complexes play a critical role in this process by modifying the structure of chromatin to make it more accessible to the transcription machinery. Some important chromatin remodeling complexes include SWI-SNF. These complexes use energy from ATP hydrolysis to alter nucleosome positioning, making DNA more accessible to RNA polymerase. ISWI. This complex is involved in nucleosome spacing and plays a role in chromatin remodeling during transcription initiation. NERD. This complex plays a dual role in gene repression and chromatin remodeling. 6. Co-activators and company repressors. In addition to the core transcription factors, co-activators and co-repressors play essential roles in fine-tuning transcriptional regulation. These proteins do not directly bind to DNA but work by interacting with transcription factors and modifying the chromatin environment or RNA polymerase activity. Co-activators. These proteins enhance transcriptional activity by facilitating the binding of RNA polymerase to the promoter or by promoting chromatin decondensation. CBP-P300 and mediator are examples of co-activators. Co-repressors. These inhibit transcription by recruiting histone deacetylases, HDACs, or chromatin-modifying enzymes that compact chromatin and prevent transcription. NCOR and SMRT are examples of co-repressors. 7. Post-translational modifications and regulation. Transcription factors and components of the transcription machinery are often regulated by post-translational modifications, PTMs, such as phosphorylation, acetylation, methylation, ubiquitination, and sumolation. These modifications alter the activity, stability, and interactions of transcription factors, providing an additional layer of regulation. For example, phosphorylation of RNA polymerase II at the CTD is a critical step in transcription initiation, elongation, and termination. Acetylation of histones opens up chromatin structure, allowing easier access for the transcription machinery. These modifications are dynamic and can be influenced by various signaling pathways, enabling cells to respond to environmental stimuli or developmental cues. 8. Previous year questions. Question. CSIR-NET 2022. Which of the following general transcription factors is involved in the formation of the pre-initiation complex PIC in eukaryotic cells? A. TFIIH. B. TFIID. C. TFIIE. D. TFIV. Answer: B. TFID. TFIID is responsible for recognizing the TATA box and is the first factor to bind the promoter region to initiate the formation of the pre-initiation complex. Question SciRNET 2021. What is the primary function of the mediator complex in eukaryotic transcription? A. It binds directly to the TATA box. B. It bridges the interaction between regulatory transcription factors and RNA polymerase II. C. It helps with chromatin remodeling. D. It synthesizes mRNA. Answer: B. It bridges the interaction between regulatory transcription factors and RNA polymerase II. The mediator complex helps coordinate transcriptional activation by interacting with both regulatory proteins and the RNA polymerase complex. Question CSIRNET 2020. Which of the following general transcription factors is responsible for helicase activity and DNA unwinding during transcription initiation? A. TP. B. TFIIH. C. TFIIF. D. TFIB. Answer: B. TFIIH. TFIIH possesses helicase activity and is essential for unwinding the DNA during transcription initiation. Conclusion. The transcription factor machinery plays a crucial role in regulating gene expression, ensuring that transcription occurs precisely and at the right time. By understanding the components of this machinery, including RNA polymerase II, GTFs, co-activators, and chromatin remodeling complexes, you can grasp the complexity of transcriptional regulation in eukaryotic cells. This knowledge is essential for answering questions in the CSIRNet JRF Life Sciences exam related to gene expression and regulation. Let me know if you would like further elaboration on any part of this topic. You said, formation of initiation complex. ChatGPT said, formation of the initiation complex. CSIRNet JRF Life Sciences. The formation of the initiation complex is a critical step in the transcription process, enabling RNA polymerase to initiate transcription at the correct location on the DNA template. In eukaryotic cells, this process is complex and requires the assembly of a variety of proteins, including general transcription factors, GTFs, RNA polymerase II, and other cofactors. The initiation complex must be precisely formed to ensure that transcription occurs at the correct promoter and that RNA synthesis proceeds efficiently. This lecture will provide a detailed explanation of the formation of the initiation complex, with an emphasis on the various component steps involved. The following points will be discussed. Overview of the initiation complex. General transcription factors, GTFs. Formation of the pre-initiation complex, PIC. Role of mediator complex and company activators. Chromatin remodeling. Phosphorylation of RNA polymerase II. Transition to elongation. Previous year questions. 1. Overview of the initiation complex. The initiation complex is a multi-protein complex assembled at the promoter region of a gene, allowing RNA polymerase II to initiate transcription. It includes the general transcription factors, GTFs, RNA polymerase II, and additional regulatory factors. This complex facilitates the unwinding of DNA, the formation of the transcription bubble, and the start of RNA synthesis. The steps involved in the formation of the initiation complex can be broken down into two main phases. Assembly of the pre-initiation complex, PIC. Activation of RNA polymerase II and transition to elongation. 2. General transcription factors, GTFs. The formation of the initiation complex in eukaryotes depends heavily on the general transcription factors, GTFs, which are required by RNA polymerase II for transcription initiation. These factors are recruited to the core promoter region of the gene, typically located upstream of the transcription start site. The main GTFs involved in the formation of the initiation complex include TFIID. This is the first transcription factor to bind to the DNA. The TBP TATA binding protein subunit of TFIID binds to the TATA box, a conserved sequence found in many promoters. TFIID also includes several TAFs TBP associated factors that help stabilize the binding of TBP to the promoter and further recruit other GTFs. TFIIA. This factor stabilizes the interaction between TFIID and the DNA. It helps prevent the dissociation of TFIID from the promoter and recruits other transcription factors. TFIIB. After TFIID binds to the promoter, TFIIB is recruited. It binds to both TFIID and RNA polymerase II, positioning the polymerase at the transcription start site. TFIIE. This factor recruits TFIIH, which is responsible for unwinding the DNA at the transcription start site, allowing RNA polymerase to access the DNA template. TFIIH. TFIIH has two key functions. It acts as a helicase to unwind the DNA and it phosphorylates the C-terminal domain, CTD, of RNA polymerase II, a critical step that triggers the transition from initiation to elongation. 3. Formation of the pre-initiation complex, PIC. The pre-initiation complex, PIC, is the assembly of RNA polymerase II and the general transcription factors of the promoter. The process begins with the binding of TFIID to the TATA box in the promoter region. This is followed by the sequential recruitment of other GTFs. TFIID binds to the TATA box, forming the first stable protein DNA interaction. TFIIA binds to the complex, stabilizing TP's interaction with the DNA. TFIIB is recruited, which helps guide RNA polymerase II to the correct location at the promoter. RNA polymerase II then binds to the complex with the help of TFIIF. TFIIE and TFIIH are the last factors to be recruited. TFIIE helps recruit TFIIH, which has helicase activity, unwinding the DNA around the transcription start site. Once all these factors are assembled, the PIC is formed, and the RNA polymerase is positioned at the transcription start site. 4. Role of mediator complex and company activators. The mediator complex is a multi-protein complex that bridges the interaction between regulatory transcription factors, activators, and the core transcription machinery. While it is not directly involved in the assembly of the PIC, the mediator complex is crucial for the activation of transcription. It facilitates the interaction between enhancer-bound transcription factors and the pre-initiation complex. Additionally, co-activators like CBP-P300 are recruited to the promoter region by transcription factors, assisting in chromatin remodeling and promoting transcriptional activation by modifying the chromatin structure. Co-activators can acetylate histones, leading to a more open chromatin configuration and facilitating access to the transcription machinery. 5. Chromatin remodeling. Chromatin structure must be modified to allow the transcription machinery access to the DNA template. Chromatin remodeling complexes, such as SWI, SNF, and ISWI, help move nucleosomes away from the transcription start site or modify histones to create a more open chromatin structure. These complexes use ATP hydrolysis to alter the positioning of nucleosomes, making the DNA more accessible to RNA polymerase II and GTFs. 6. Phosphorylation of RNA polymerase II. Once the PIC is assembled, TFIIH phosphorylates the C-terminal domain, CTD, of RNA polymerase II. This phosphorylation is a key regulatory step in the initiation process. The phosphorylation of the CTD enables the transition from transcription to initiate elongation. It also serves as a docking site for other factors involved in RNA processing, such as capping and splicing enzymes. 7. Transition to elongation. After the initiation complex is assembled and transcription begins, the RNA polymerase moves along the DNA template, synthesizing RNA in the 5' to 3' direction. As RNA polymerase II synthesizes the RNA, it is phosphorylated at specific residues, which helps transition from initiation to elongation. The phosphorylation of the CTD serves as a signal for the recruitment of additional elongation factors, and the RNA polymerase leaves the promoter region to proceed with RNA synthesis. 8. Previous year questions. Question. CSIRNET 2021. Which of the following transcription factors is directly responsible for the unwinding of DNA during transcription initiation? A. TFIIH. B. TFIIB. C. TFIID. D. TP. Answer. A. TFIIH. TFIIH contains helicase activity, which unwinds the DNA during transcription initiation. Question. CSIRNET 2020. Which component of the transcription initiation complex is involved in the phosphorylation of RNA polymerase II, signaling the transition to elongation? A. TP. B. TFIIH. C. TP. D. Mediator complex. Answer. B. TFIIH. TFIIH phosphorylates the C-terminal domain CTD of RNA polymerase II, allowing the transition to elongation. Question. CSIRNET 2019. What is the primary function of the mediator complex in the initiation complex? A. It binds the ta-ta box. B. It bridges the interaction between transcription factors and RNA polymerase II. C. It is responsible for DNA unwinding. D. It phosphorylates the CTD of RNA polymerase II. Answer. B. Mediator complex. The mediator complex bridges the interaction between transcription factors and RNA polymerase II. Conclusion. The formation of the initiation complex is a highly coordinated process involving the assembly of RNA polymerase II, general transcription factors, chromatin remodeling complexes, and co-activators of the gene promoter. Understanding the roles of these components, as well as their interactions, is essential for understanding transcriptional regulation and the initiation of gene expression. This process is a fundamental concept in molecular biology and is often tested in the CSIRNet JRF Life Sciences exam. You said. Transcription activator ChatGPT said. Transcription activators CSIRNet JRF Life Sciences. Transcription activators are specialized regulatory proteins that bind to specific DNA sequences, often enhancers of upstream activating sequences, and significantly enhance the transcription of target genes. They are vital for the regulation of gene expression, acting as switches that determine whether a gene should be turned on or off, depending on cellular signals, environmental stimuli, or developmental stage. These proteins do not directly bind RNA polymerase but act through protein-protein interactions and chromatin modifications to facilitate the assembly and function of the transcriptional machinery. In eukaryotic systems, where chromatin structure poses an additional barrier to transcription, transcription activators are essential to making DNA accessible and recruiting the transcriptional machinery to the correct gene. They are typically modular in nature, with distinct domains responsible for DNA binding and activation. This lecture covers the following key areas. Structure of transcription activators. DNA binding domains and activation domains. Mechanism of action of activators. Role in chromatin remodeling. Interaction with coactivators and mediator. Enhancers and activator binding sites. Examples of transcription activators. Previous year questions with answers. 1. Structure of transcription activators. Transcription activators generally consist of two functional domains. DNA binding domain (DBD). This domain recognizes and binds to specific DNA sequences, enhancers of upstream regulatory elements. Activation domain (AD). This domain interacts with coactivators, chromatin remodeling complexes, or general transcription factors to enhance transcription. Some activators may have additional domains for signal sensing, e.g., ligand binding domain and nuclear receptors dimerization, or regulatory control. 2. DNA binding domains and activation domains. Common DNA binding domains include helix turn helix HTH found in homeodomain proteins. Zinc finger motifs seen in steroid hormone receptors. Leucine zipper BZIP found in proteins like CREB. Helix loop helix VHLH found in transcription factors such as MYD. Activation domains. Acidic activation domains found in yeast activators like GAL4 and BP16. Glutamine rich domains found in SP1. Proline rich domains found in CTF and F1. These domains do not directly contact the basal transcription machinery but work through protein-protein interactions. 3. Mechanism of action of activators. Transcription activators increase gene expression by several mechanisms. Recruitment of coactivators. They bind coactivators like CBP P300 which possess histone acetyltransferase activity, loosening chromatin. Interaction with the mediator complex. Activators help recruit the mediator, which bridges activators and RNA polymerase II. Stabilization of the pre-initiation complex picked by recruiting GTFs and RNA pol II to the promoter. Chromatin modification. Indirectly promote transcription by altering histone structure through recruitment of remodeling complexes. Activators may work synergistically, where two or more activators together lead to much higher transcription than each alone. 4. Role in chromatin remodeling. DNA is packed into chromatin, which can repress transcription. Activators recruit chromatin remodeling complexes like SWI-SNF, which use ATP to reposition nucleosomes, making DNA accessible. They also recruit histone acetyltransferases (HATs) to acetylate histones and reduce their affinity for DNA, making the promoter region more open. 5. Interaction with coactivators and mediator. Coactivators do not bind DNA directly but are recruited by activators. Some key coactivators include CBP P300, histone acetyltransferases that modify chromatin to an open state. Mediator complex, a multi-subunit complex that physically links activators with RNA polymerase II and GTFs. Mediator acts as an integrator of signals and assists in the formation and stabilization of the transcription initiation complex. 6. Enhancers and activator binding sites. Activators bind to enhancers, which may be located upstream, downstream, or even within the genes they regulate. These enhancers work irrespective of orientation or distance due to DNA looping, allowing activators to interact with the promoter-bound transcription machinery. Enhancer-promoter communication is facilitated by the looping of DNA, bringing distant regulatory elements in close proximity with the core transcription machinery. 7. Examples of transcription activators. GAL4 East recognizes UASG sequences, activates transcription via acidic activation domain. NF-kappa B activates immune and inflammatory genes upon stimulation. CREB, activated by phosphorylation via CAMP signaling, binds to CAMP response elements. P53, a tumor suppressor that activates genes involved in DNA repair, apoptosis, and cell cycle arrest. Estrogen receptor ER, activated by hormone binding, binds to estrogen response elements ARES. These transcription activators often respond to external signals and integrate intracellular pathways to fine-tune gene expression. 8. Previous year questions with answers. Question. CSIRNET 2022. Which of the following is true regarding transcriptional activators in eukaryotes? A. They bind RNA polymerase II directly at the core promoter. B. They bind to ta-ta box in the promoter region. C. They bind to enhancer regions and facilitate transcription via coactivators. D. They unwind DNA at the transcription start site. Answer. C. They bind to enhancer regions and facilitate transcription via coactivators. Question. CSIRNET 2020. Which of the following is a function of the acidic activation domain of a transcriptional activator like GAL4? A. DNA methylation. B. Direct RNA synthesis. C. Histone acetylation via recruitment of coactivators. D. DNA replication. Answer. C. Histone acetylation via recruitment of coactivators. Question CSIRNET 2019. Which of the following statements about transcription activators is incorrect? A. They bind to specific DNA sequences. B. They interact with the basal transcription machinery. C. They have histone methyltransferase activity. D. They often possess acidic or proline-rich activation domains. Answer. C. They have histone methyltransferase activity. Correct explanation. Transcription activators themselves do not have histone methyltransferase activity. They may recruit such enzymes indirectly. Conclusion. Transcription activators are key regulators of gene expression in eukaryotic cells. They perform their function by binding specific regulatory sequences, recruiting coactivators and chromatin remodeling complexes, and facilitating the assembly and activation of the transcription machinery. Their modular architecture allows flexibility and adaptability in regulation. Understanding their mechanisms, domains, and interactions is essential for cracking CSIRNet JRF life sciences, as this area is frequently tested and holds significant conceptual weight. You said. Transcription repressor. ChatGPT said. Transcription repressors. CSIRNet JRF life sciences. Detailed lecture. Maximum detail. No table PYQs included. Transcription repressors are regulatory proteins that inhibit gene expression by interfering with the process of transcription. While transcription activators promote transcription by enhancing the recruitment and function of RNA polymerase and general transcription factors, repressors prevent transcription through multiple molecular strategies. These mechanisms are crucial for maintaining cell type specificity, temporal gene expression, developmental control, and response to environmental signals. Understanding transcription repressors is essential for the CSIRNet JRF life sciences exam, especially given that questions often test knowledge of gene regulatory mechanisms, protein DNA interactions, chromatin structure modulation, and the function of specific repressor pathways. 1. Definition and importance. A transcription repressor is a DNA binding protein that negatively regulates gene expression by blocking the transcription of target genes. It binds to specific DNA sequences, often overlapping or adjacent to the promoter or enhancer regions, thereby preventing the access or function of the transcriptional machinery. This ensures that genes are expressed only when required, contributing to energy conservation and regulatory precision. 2. Types of transcription repressors. Repressors can be classified based on their mode of action. A. Competitive repressors. They bind to DNA sequences that overlap with or are adjacent to activator binding sites, thereby competing with activators for binding. This direct competition prevents the recruitment of coactivators or general transcription factors. B. Masking repressors. Quenching. They do not block DNA binding of activators but bind directly to the activation domains of activators, thereby preventing them from interacting with the transcriptional machinery or coactivators. C. Direct repression through core repressor recruitment. They recruit core repressor complexes such as histone deacetylases, HDACs, histone methyltransferases, or other chromatin-modifying enzymes, which compact chromatin and make it inaccessible for transcription initiation. D. Repressor-mediated chromatin condensation. They may actively recruit chromatin remodeling complexes that compact nucleosomes and silence gene expression, such as polycomb group proteins PCG. 3. Mechanisms of action. Blocking RNA polymerase recruitment. Repressors can bind to operator or silencer regions near the promoter to prevent the assembly of the pre-initiation complex PIC or block the recruitment of RNA polymerase II. E. Preventing enhancer-promoter communication. By binding to insulator elements or silencing elements, repressors can prevent looping of DNA, thereby disrupting enhancer-promoter communication required for transcription activation. E. Modulating chromatin state. Repressors recruit chromatin-modifying enzymes. Histone deacetylases HDACs remove acyl groups from histones, causing tighter DNA histone interaction and transcriptional repression. Histone methyltransferases HMTs methylate histones to generate repressive marks like H3K9me3 or H3K27me3, which are associated with heterochromatin. Nucleosome remodeling complexes compact chromatin or reposition nucleosomes to cover the transcription start site. 4. Silencer elements. Silencers are specific DNA sequences to which repressors bind. These elements can act at a distance and in an orientation-independent manner, much like enhancers. When bound by repressors, they inhibit transcription by preventing chromatin opening, PIC assembly, or enhancer function. 5. Examples of transcription repressors like REST and RSF, Re1 silencing transcription factor, represses neuronal genes in non-neuronal tissues by recruiting chorus and HDACs. E. MAD-MAX complex. Opposes MYC-MAX activation by recruiting SYN3 core repressor and HDACs. E. Groucho TLE family, in Drosophila invertebrates, recruited by repressor proteins to mediate gene silencing during development. IB, snail and slug, zinc finger repressors. Suppress E-cadherin expression during epithelial to mesenchymal transition, EMT, essential in development and cancer metastasis. B. Polycomb repressive complexes, PRC1 and PRC2, epigenetically silence genes through H3K27 methylation and chromatin compaction. 6. Transcriptional core repressors. Core repressors are non-DNA binding proteins that are recruited by repressors to execute repression. Examples include NCOR and SMRT, core repressors of nuclear hormone receptors that recruit HDACs. SYN3A, recruited by many repressors including MAD, REST, and MECP2. CTBP, C-terminal binding protein, interacts with many repressor proteins and brings HDACs and other chromatin modifiers. These multiprotein complexes often form large repressive assemblies on chromatin to silence transcription stably inheritably. 7. Biological significance. Developmental regulation ensures genes are turned off in inappropriate tissues, e.g., REST represses neuronal genes in non-neuronal tissues. Cell cycle control. Repressors like RB protein block expression of S-phase genes. X chromosome inactivation, mediated by long non-coding RNAs and chromatin-associated repressor complexes. Cancer biology. Mutations in repressors like p53 or deregulation of repressive chromatin marks are linked to tumorigenesis. 8. Previous year questions with answers 1. CSIRNet Life Sciences 2021. Which of the following is a true mechanism by which a transcriptional repressor can inhibit gene expression? A direct methylation of the promoter DNA. B. Activation of histone acetyltransferase activity. C. Binding to enhancer elements to facilitate activator function. D. Recruitment of histone deacetylases to the promoter region. Answer. D. Recruitment of histone deacetylases to the promoter region. Explanation. This is a classical mechanism of repression wherein HDACs remove acyl groups, leading to chromatin condensation and transcriptional repression. 2. CSIRNet Life Sciences 2019. REST Re1 silencing transcription factor represses neuronal gene expression in non-neuronal cells by activating transcription factor CREBB, recruiting coactivators to histone acetylate nucleosomes. C. Binding to promoter and recruiting histone deacetylases. D. Promoting enhancer promoter communication. Answer. C. Binding to promoter and recruiting histone deacetylases. Explanation. REST binds to Re1 sites and recruits chorus and HDACs to silence neuronal genes outside the nervous system. 3. CSIRNet Life Sciences 2020. Which of the following protein complexes is associated with transcriptional repression? A. SWI-SNFB mediator CCBP P300. D. Polycomb repressive complex. Answer. D. Polycomb repressive complex. Explanation. PRC1 and PRC2 are known to repress gene expression by methylating H3K27 and compacting chromatin. Conclusion. Transcription repressors are indispensable to the precise control of gene expression, acting through DNA binding competition, masking activators, recruiting co-repressors, and remodeling chromatin to a repressive state. Their roles span development, differentiation, metabolism, and disease, making them high-priority targets in transcriptional regulation questions for CSIR-NET JRF Life Sciences. A strong grasp of their mechanisms, examples, and differences from activators is crucial for cracking conceptual and applied questions in the exam. You said, RNA polymerases. ChatGPT said, RNA polymerases. CSIR-NET JRF Life Sciences. Detailed theory lecture. Maximum detail based on PYQ areas. No table includes PYQs with year and answers. RNA polymerases are essential enzymes responsible for the synthesis of RNA from a DNA template in a process known as transcription. These enzymes read the DNA template strand in the 3' to 5' direction and synthesize a complementary RNA strand in the 5' to 3' direction. In both prokaryotes and eukaryotes, RNA polymerases are central to gene expression, but their structure, number, and functions differ significantly between the two domains. Understanding RNA polymerases is crucial for the CSIR-NET JRF Life Sciences exam because transcription is a fundamental process in molecular biology, and questions frequently test candidates on types of polymerases, their roles, inhibitors, subunit composition, promoter recognition, and associated factors. 1. RNA polymerase in prokaryotes. Prokaryotic organisms, such as Escherichia coli, have a single type of RNA polymerase responsible for transcribing all classes of RNA, mRNA, tRNA, and rRNA. Structure. Prokaryotic RNA polymerase is a multi-subunit enzyme. The core enzyme has 5 subunits. Alpha 2 copies involved in enzyme assembly and interaction with regulatory proteins. Beta, catalytic subunit responsible for RNA synthesis. Beta binds to the DNA template. Omega assists in enzyme assembly and stability. To initiate transcription, the sigma factor joins the core enzyme to form the holoenzyme, which can recognize and bind specific promoter sequences. Function of sigma factor. It directs the holoenzyme to the promoter by recognizing conserved sequences like the -10 prit box and -3 regions. After initiation, the sigma factor dissociates, and the core enzyme proceeds with elongation. Different sigma factors recognize different classes of genes under various conditions, providing regulatory specificity. 2. RNA polymerases in eukaryotes. Eukaryotic cells contain 3 primary nuclear RNA polymerases, each with distinct roles. A RNA polymerase I. coli, located in the nucleolus, transcribes a single gene type, the large rRNA precursor 45S, which is processed into 18S, 5.8S, and 28S rRNAs. B. RNA polymerase II, Pol II, located in the nucleoplasm, transcribes protein coding genes mRNA and also synthesizes snRNAs in some RNAs. Requires several general transcription factors GTFs for initiation, e.g., TFIID, TFIIH. Associated with CTD, C-terminal domain of the largest subunit RPB1, which undergoes phosphorylation to regulate initiation and elongation. C. RNA polymerase III, Pol III, located in the nucleoplasm, transcribes tRNAs, 5S rRNA, and other small RNAs. Each of these polymerases recognizes different promoter elements and utilizes distinct transcription factors to initiate transcription. 3. Mitochondrial and chloroplast RNA polymerases. These organelles possess bacteria-like single subunit RNA polymerases that are similar to T7 bacteriophage polymerase. They transcribe mitochondrial or chloroplast genes and are nuclear encoded but function within the organelle. 4. RNA polymerase structure and catalytic mechanism. RNA polymerases share a conserved catalytic core characterized by a crab claw-like shape. The enzyme forms a transcription bubble, approximately 17 base pairs, by unwinding DNA. Nucleoside triphosphates (NTPs) are added one by one to the 3' of the grown RNA chain, and pyrophosphate is released with each addition. The enzyme progresses with high processivity and fidelity, although proofreading is limited compared to DNA polymerases. The Mg² ion is essential for catalysis, coordinating the phosphate groups of NTPs. 5. Inhibitors of RNA polymerases. Actinomycin D intercalates DNA and blocks elongation by all polymerases. Rifampicin specifically inhibits bacterial RNA polymerase by binding the beta subunit and preventing transcription initiation. Used against Mycobacterium tuberculosis. Alpha ammonidin, a toxin from Amanita phalloides mushroom, inhibits eukaryotic RNA polymerase II at low concentrations and RNA polymerase III at higher doses. Poli is resistant. These inhibitors are often exam favorites in CSIR-NET due to their specificity and practical applications in research and medicine. 6. Regulation of polymerase activity. RNA polymerase activity is modulated by transcription factors, chromatin structure, post-translational modifications, especially CTD phosphorylation in Pol II, and interactions with enhancers silencers. The transition from initiation to elongation in Pol II is a highly regulated step involving kinases like TFIIH, which phosphorylates CTD, promoting promoter clearance. 7. Differences between RNA polymerase and DNA polymerase. RNA polymerase does not require a primer. It can initiate synthesis de novo. It lacks exonuclease proofreading activity, making RNA more error-prone. DNA polymerases require a template and primer, and they have higher fidelity with proofreading. Previous year questions with answers 1. CSIR-NET Life Sciences 2018. Which one of the following toxins specifically inhibits eukaryotic RNA polymerase II? A. Rifampicin B. Actinomycin D. C. Alpha ammonidin D. Cyclohexamide. Answer. C. Alpha ammonidin. Explanation. Alpha ammonidin inhibits RNA polymerase II at nanomolar concentrations, making it a potent tool in transcription studies and a common exam question. 2. CSIR-NET Life Sciences 2020. In eukaryotic cells, which RNA polymerase is responsible for the transcription of tRNA genes? A. RNA polymerase IB. RNA polymerase II. Circa RNA polymerase III. D. Mitochondrial RNA polymerase. Answer. C. RNA polymerase III. Explanation. RNA Pol III transcribes tRNAs, 5S rRNA, and small nuclear RNAs. 3. CSIR-NET Life Sciences 2021. Which subunit of bacterial RNA polymerase recognizes the promoter region of DNA? A. Beta B beta, C alpha D sigma. Answer. D sigma. Explanation. The sigma factor is essential for promoter recognition and binding in prokaryotes. It is released after initiation. Conclusion. RNA polymerases are central to the gene expression machinery in all living organisms. Prokaryotes utilize a single enzyme with sigma factors for specificity, whereas eukaryotes possess 3 distinct polymerases, poli, 2, 3 each dedicated to transcribing specific types of genes. Understanding their structure, function, specificity, and inhibitors is critical for the CSIR-NET JRF Life Sciences exam. Mastery of this topic ensures clarity in transcriptional regulation and builds a solid foundation for related topics like transcription factors, initiation complex formation, and RNA processing. You said, capping. ChatGPT said, capping. CSIR-NET JRF Life Sciences. Complete theory lecture. Maximum detail based on previous year questions. No table includes PYQs with year and answers. Capping is a vital co-transcriptional modification that occurs at the 5' end of nascent free mRNA transcripts in eukaryotic cells. This modification results in the formation of a unique structure known as the 5' cap, which is essential for mRNA stability, nuclear export, efficient translation, and protection from exonucleases. The process of capping occurs almost immediately after the RNA polymerase II transcribes around 20 to 30 nucleotides of the mRNA. It is an indispensable quality control and regulatory step in eukaryotic gene expression and is not seen in prokaryotes. 1. Definition and structure of the 5' cap. The 5' cap is a modified guanine nucleotide attached to the first nucleotide of the RNA transcript via 5' to 5' triphosphate linkage, which is chemically unusual and resistant to nucleases. This cap is further methylated at the N7 position of the guanine base, resulting in 7-methylguanosine M to the power of 7G. In some cases, the ribose sugars of the first and second nucleotides are also methylated at the 2' position, forming cap 1 and cap 2 structures respectively. 2. Steps in the capping process. Capping involves 3 major enzymatic steps that occur co-transcriptionally. I. RNA triphosphatase activity. The enzyme removes the gamma phosphate from the 5' triphosphate end of the nascent RNA, leaving a diphosphate end. E. Gangliotransferase activity. This enzyme adds GMP, guanosine monophosphate, from GTP to the diphosphate end of the RNA through a 5' to 5' triphosphate linkage, creating the basic cap structure. E. Methyltransferase activity. The enzyme methylates the guanine at the N7 position, converting the cap to 7-methylguanosine M to the power of 7G, which is a hallmark of a functional 5' cap. Additional methylations may occur on the ribose sugars of the first and second nucleotides. 3. Enzyme complex and CTD involvement. The enzymes involved in capping are recruited by the phosphorylated C-terminal domain (CTD) of RNA polymerase II. Specifically, the SR5 phosphorylated CTD serves as a platform for assembling and positioning the capping machinery. This coordination ensures that capping is tightly coupled with transcription. 4. Biological functions of the 5' cap of protection from degradation. The 5' cap protects free mRNA from degradation by 5' exonucleases, increasing transcript stability. B. Facilitates nuclear export. Capping is required for the binding of cap binding complex (CBC), which mediates nuclear export of mRNA through the nuclear pore complex. C. Promotes translation initiation. In the cytoplasm, the cap is recognized by A4E, a component of the eukaryotic initiation factor.